I really appreciate the invitation. My approach on this is to try to make sure that my perception of the scientific findings, both pro and con with respect to genetic engineering, are presented. So that's what I'm going to try to do, and I'm going to make sure we have plenty of time for questions. First, it's very important that I start off with a list of all the industry, private sector funding that I have received on genetic engineering, research, and outreach. And that's the list right there. So I'm actually not a molecular biologist. I've done some molecular biology. We have molecular biologists in the audience. So that gives me a bit of a distance from the issue. I respect my colleagues very much, respect what they say, but I have none of this. And so I think that I always like to make sure people know that. But these people are my teachers, so I really value them. So genetic engineering is already part of our lives in big ways. If you know a diabetic, they are very likely using insulin that is from a genetically engineered microorganism, and as such, it's higher quality than what you could get from extractions from the pancreas of animals. The enzymes for hard cheese are very commonly genetically engineered, and I don't think there's an advanced cancer therapy in the world that doesn't depend somewhere in the process of research development and commercialization and clinical use that doesn't have a component of genetic engineering. It's just part of biology nowadays. So I want to talk a little bit about what genetic engineering is, which is another way. GMOs is commonly what the public says, genetically modified organisms. And by that, what I think the public means is genetic engineering, which is the phrase that most scientists prefer, because genetic modification happens every generation in every species. It's not, genetic modification is a very loose term, whereas genetic engineering describes what we do in the lab, whether you approve or disagree. So genetic engineering is the phrase I think that fits for scientists. First of all, how many have heard of Roundup Ready crops? Okay, in this audience, I'm not surprised to see so many hands. So I want to say that there are two aspects of this that are very important. One, Roundup Ready or engineered or herbicide-tolerant crops, even those derived from conventional breeding, is useful but not sustainable. It's useful but not sustainable. So whatever concerns you have about Roundup Ready crops, let's take them off the table, and we can talk about those as much as you want. But that is not the model of genetic engineering that I think is important. Okay, I forgot to get an apple, but normally what I would do with this slide is take a bite of an apple, and I would chew, and I would say, now how many, raise your hand if you think I'm eating DNA. So if I had an apple in my mouth, raise your hand, would I be eating DNA? Raise your hand if you think so. Okay, so again, this is a biologically savvy crowd. Yes, I'm eating DNA. I'm eating DNA of an apple. I'm eating DNA of bacteria on the surface of the apple, fungi, probably some yeasts, and maybe some insect eggs. So lots of DNA would be in my mouth, and it's all safe. DNA has been recognized for decades and decades as a safe substance to eat. Everybody's laughing. I wonder why. Okay, somebody explain to me later, okay? All right, so DNA is in all our food. It's a natural part of our diet. Okay, so I want to start with that. Doesn't mean you can't make a dangerous crop with genetic engineering, but what I'm saying is DNA itself is common, it's abundant, and it's part of our normal diet. And I am talking about this part before we get to Boxwood. DNA, biologists, I'm going to represent all biologists here, or most. Biologists view DNA as chemical information. It's not a mysterious substance, it's just chemical information like words, letters in a book. And genes would be the sentences in the book, okay? That's the way we think about it, and this, I think this has been, I've never had a biologist refute this kind of fundamental way of thinking. So genetic change occurs, and it occurs in one of three very broad ways. First of all, let's not forget that nature is doing it all the time, in crazy ways, in bizarre ways. Nature creates genetic changes. There is conventional breeding, so breeding is, there better be genetic change if you're going to breed, right? Because you need genetic variation to do a breeding program. So that's another way that genetic change occurs, and then genetic engineering, which I'll explain again. So let's start with nature. If you eat sweet potatoes, you are assuredly eating not only the genetics of the plant, well, the genetics of the plant, maybe it's better to say, the genetics of sweet potato include genetics of bacteria as part of their genetics, not bacterial cells. I mean, the bacterial DNA has been incorporated by nature into sweet potato. And as bizarre as that sounds, that's well established. It was published a few years ago. So nature does crazy things. And I mean, it just astounds me what nature does creatively with genetics. And I won't say any more for lack of time. Whenever we do breeding, we do genetic modification. That is very familiar to you. On the bottom, the plant, the ear on the top is actually the progenitor of this which we recognize as corn. And humans selected those genetic modifications that were occurring naturally. Okay, so genetic modification occurs all the time, and we're glad for it, or at least I am. And when we do conventional breeding, crazy things do happen with genetics. They just, again, I read the scientific literature, and I was like, my gosh, wow, what crazy things nature does. So hybridization, especially between species, creates all kinds of unexpected and uncharacterized genetic changes. This is called protoplast fusion, where you strip the cell walls off of plant cells in liquid media and you smash them together and create new species that don't exist in nature. And that is a conventional breeding technique. That is not genetic engineering. And so that would fly under everybody's radar. And so it sounds like I'm a cheerleader for genetic engineering. On the one hand, I am. We're going to talk about the limitations too, because they belong in this talk too. This is a conventional technique. So that guy is handling a very hazardous mutagenic chemical. And he's going out there and painting it on the silks of these corn plants to create mutations, lots of them, that he has not characterized. And the result is to hopefully create a better corn. That's conventional breeding. That flies under the radar. So I said there was a third type of genetic change, genetic engineering. So let me give you a concept of what that involves. I think the best way to understand it is like what you do with your word processor. When you copy a sentence out of a file and you paste that sentence into another file, that's kind of what genetic engineering is. It's you're selecting a certain target, copying it, and pasting it into another organism. That's really the concept. And another concept that's important to understand as you either evaluate or critique genetic engineering is the concept of gene pool. So we all understand that dogs can interbreed, but they can't breed with cats. So the gene pool of dogs is dogs, right? And I guess maybe wolves, maybe. I'm not sure if they can breed with wolves at this point, but there's a certain restricted pool of organisms with which they can hybridize, and that's the gene pool. Okay, so when we talk about genetic engineering, we may be talking about taking genes from way outside the gene pool. Okay, so a corn gene into an apple is outside the gene pool, and we can only do that in the lab. Or we could take even a gene from a bacterium, okay, and insert that into an apple, and we can do that, and generally it's going to function. And so that's genetic engineering. Or we can engineer with something that's within the gene pool, genetics within the gene pool, and that goes by a different name. On the left, it's transgene. On the right, it's cisgene. And if you're working with cisgenes, you're still within the breeding pool, so theoretically that transfer could occur by natural means, but it may take 20 or 50 or 100 years to get the combination you want, so you do it in the lab because of time. Okay, so how does this actually work? So we're talking still about genetic engineering. So let's say, I'm of Italian descent, so I like Italian food. So let's say I'm writing a book on Italian food, and I have the introduction and the history section and the recipes. And this sentence right here, I want to put in the book. Tomato was added to the focaccia in the late 18th century. So that might fit very well in the history section. So one way that I can insert a gene into another organism is by what's called plant transformation. So it's a copy and paste mechanism, okay? And so there you can see a recipe, and As luck would have it, this insertion, because I can do lots, but this particular insertion inserted right there. So now we've got the sentence we want, but it's disrupted a gene, right? A part of a recipe, right? So we'll call that a gene, right? So that's that may not be desirable if that recipe is particularly important, but at least we got the gene that we wanted in there. And that's the older technique called plant transformation, which still is useful, but that's the downside, is this random insertion. So you'll do lots of these, hoping to get some that are useful. I like this picture because that's her plant transformation machine right there. And genetic engineering, I like to say, is done by real people. I mean, these are not, wild-eyed scientists. They're just people like us. And she's very proud, got that big smile on her face. And so anyway, she's a good-hearted person, and I wanted to put that in there. So the other way that I think I've only put in 2 for this talk, but the other way of genetic engineering is by gene editing. This is not plant transformation. This is gene editing, or CRISPR. Who's heard of CRISPR? I'll bet a lot of hands. Yep. Okay, good. So this is the fundamental concept of CRISPR, is you can make targeted edits, just like with your word processor. Okay, so how does that look? So here we have a section out of the history portion of the book. The precursor of pizza was probably the focaccia, a flatbread known to the Romans as panis focaccius, to which toppings were then added. And I like that sentence, but I want to put in that sentence about tomato being added right there. Well, with CRISPR, you largely can. Okay, and this is one of the reasons why CRISPR is revolutionizing biology. Yeah, because it gives a high degree of precision to do genetic change. So here's another way CRISPR could be useful. So wine is made from grape juice. That's wrong, right? There's a misspelling, but just one letter. So we can't fix that letter with transformation, but we can fix it with CRISPR. Or maybe the gene says wine is made from grapem juice. So we want to delete that misspelling so we can, we're getting better and better at being able to selectively delete or change even single letters in DNA. So very targeted edits. Now why would we even want to change single letters? Here we have a gene from wheat, a portion of a gene, that's the DNA sequence. And then the 2 on the bottom here are actually the same gene from 2 plants that are identical to this plant, except they were engineered to have these very, very modest deletions, 1 or 3 letters. Now they didn't just select this gene at random, they knew what the gene did, and they said, we think if we disrupt or mutate this gene, we will create powdery mildew resistant plants. First of all, this is an incredibly modest genetic change, so modest that you cannot distinguish it from natural mutation. There would be no way to distinguish this from natural mutation. It's so modest. But as expected, there is the control, lots and lots of powdery mildew. You can't really tell in this slide. And there's the engineered plants with these very modest changes with no powdery mildew. So that's the kind of stuff that makes me think, yay, because our growers in Kentucky use pesticides to control powdery mildew in wheat sometimes. And I think there are better alternatives down the road, hopefully. So I like this because this ability to make these very targeted and modest changes is having significant impacts in medical research. And this guy, I met him, I shook his hand, and he at a conference on CRISPR, he said to the researchers in the room, he said, please hurry up. Please hurry up. And the reason he's 32, and he's a sickle cell anemia patient, so he might live to 40. And he's hoping for a fix through CRISPR. So this is a big deal, this whole CRISPR world, not just agricultural. And a lot of people are doing it. We have graduate students and postdocs doing CRISPR in labs at the University of Kentucky, and I'm sure they're doing it at Maryland and so on. So here's a list of aspects of genetic engineering that that might surprise you. But genetic engineering versus conventional breeding is actually more precise in making genetic change. And I could show you, if you're interested, email me and I'll show you some of the data. And it makes sense. If you're just trying to insert a gene instead of hybridizing all these other genes, then you're causing less genetic change, and that's what we see. It's less impactful to the plant. Genetic engineering causes less impact to the plant. And I'm defining impact formally by referring to, for the biologists in the room, referring to the transcriptome and the proteome and the metabolome. So in other words, these major measures of plant biochemical activity are more disturbed by conventional breeding. Again, it makes sense because you're trying to insert a very minimum amount of DNA with genetic engineering. So if you want to keep the parental variety of boxwood, then you're more likely to be able to do that with genetic engineering. That's not to say there aren't challenges, but you're probably more likely to do that with genetic engineering. And also, the public is uneasy about genetic engineering. So that's a big hurdle, which I'll come back to. That's a big hurdle. All right. As far as safety on the food aspect, again, this summarizes all the science I've read. If I were worried, I would not want my family eating genetically engineered foods. I don't care if they eat genetically engineered foods. I'm totally content with the science behind the safety of the process of genetic engineering. I hope they don't eat too much sugar. I hope they don't eat too much saturated fats. All the things that nutritionists know from conservative science studies. Okay, I actually think that we can use genetic engineering, and this is my professional judgment, maybe it's a bias, I don't know, but I think we can actually use genetic engineering to make safer food. And I could, again, point you to the literature, but the one that is, to me, the slam dunk is a study published last year. How many people have heard of aflatoxins? Okay, they're natural poisons among the most carcinogenic toxins known, and they're natural. And they occur in corn, they occur in peanuts, they occur in pistachios, other nuts, cottonseed. And, you know, our food system tries to keep those things out. Does a pretty good job. But in developing countries, they get exposed to more aflatoxins, and they have risks related to those. And there was the first study I ever saw that brought an infestation of aflatoxin to zero, elimination, was a study that involved genetic engineering. So I think that there's no doubt that we can incorrectly use the technology, but we can also use it to do things that are really important. This is a breast cancer study in mice, and the reduction that you see there in tumor weight, I think I said incidence before to one of the people I talked to, but that's the weight of tumors, was genetically engineered flax. So my sister passed away from breast cancer, so when I see things like this, I'm like, yeah, I'd like to see that work continue. Let's see if it can save lives. Maybe it can. Okay. Pesticides. That's not boxwood, but it's a good picture. And you know, I know fungicides really well. I've been working with them for 40 years, and I would really like to reduce the use. But if we're going to reduce pesticide use, we have to give producers alternatives. Okay. All right, disease control, genetic engineering, slam dunks in the literature. First of all, this is conventional breeding here. This conventionally bred alfalfa looks a lot healthier than that. So you don't always need genetic engineering. We don't always need it. I'm not here to say it's better than. It's just a set of options. Was the DNA modified in this variety that was conventionally bred? Yes, some heads are nodding, yes. So remember, genetic modification occurs all the time. There are few GE projects in landscape plants. I do think there is potential if you have social acceptance. And again, I'll come back to this point. So I want to show you the relationship of genetic engineering to disease control, particularly with respect to pesticide use. But the BT crops engineered for insect resistance to certain insects come from this bacterium. It produces this kind of diamond-shaped crystal, which is actually very toxic, not to humans, not to mammals, but to certain insects and caterpillar-type insects, or there can be others that attack certain beetles. And it works great, and look what happened to insecticide use in corn with genetically engineered corn. So that's a positive benefit in spite of the challenges of genetic engineering. This is in Bangladesh. Eggplant is a very important crop culturally, and you can pick out the engineered one for insect resistance. And the smallholder farm, this is being used in Bangladesh by smallholder farmers, not by big companies, and they like it because it can really dramatically reduce their insecticide use, hugely. I wrote an article about genetic engineering and disease management, so it's open access, you can read it if you wish. So I know a lot about this topic, is what I'm saying. Here were a few that I featured in that article. Rice blast, the most important disease of the most important grain in the world. A lot of fungicide used, and look at all his protection, right, on his suit, and he's got none. So I would really like to see a better alternative, and I think there are opportunities with genetic engineering that have been published. This is late blight of potato, and green crops were engineered with cis genes, probably providing an opportunity for very dramatic reductions in fungicide use. This is down in Florida. They've engineered this tomato with a single receptor gene from pepper. It's a receptor gene, so that means that all that protein does is it recognizes the infection. That's all it does. And it allows the tomato to defend itself through its own natural defenses. And I like that better than what they're doing now. Even though this is a GMO, that's what they're doing now. They're spraying copper, which is not good for the environment. It's not good for workers. American chestnut, I've got to say a little bit about that. There's the canker from American chestnut. It's just, it's basically eliminated the American chestnut as a forest tree. And the American Chestnut Foundation is doing what would be called conventional breeding for resistance to a chestnut blight. They're using backcross breeding and genomic selection techniques. And there is also a project that involved engineering of American chestnut with a gene from wheat that fights back against the fungus. And there it is. And there are dead American chestnuts that don't have that gene, and here are Chinese chestnuts that do have that gene. Oh, sorry, that are naturally somewhat resistant, okay? Okay, that's the one with the engineered wheat gene. And what does the American Chestnut Foundation say about this gene? It says they actually like it. They consider it to be another tool in our toolbox, the genetically engineered. So they actually say this is like stacking resistance genes. Let's take our genes and your gene and put them together in the forest and let them work together to control the chestnut blight. So they view it as complementary. They don't view it as competing with them in any way. They view it as complementary. Okay, a little bit on what I can tell you about boxwood blight. I'm not a boxwood expert. I'm not a boxwood blight expert. There are some who are. But I've thought about this, read enough about it that I think I can give you some guidance. Yeah, it's pretty ugly, right? All right. So disease control practices boil down to one of four general categories. Chemical, which is useful but not desirable. Cultural practices, which it's very interesting to hear about the work with mulching. We'll certainly follow that to see where it goes. Biological control is great when it works, but for diseases it's tough to get it to work. And genetics. And I like genetics. I think that there was a great talk by this gentleman from Belgium on genetic approaches. Yay, genetics. So I think my judgment is on this disease, as I've read about it, is the resistance of some form, and I don't care how, genetic engineering or conventional, resistance of some form that's genetically based, may be the best path to sustainable boxwood use in the landscape. Why resistance? So now you got the pathologist here talking. One is it's a destructive disease on a perennial crop. You got it in there, it's going to stay in there. The pathogen is easily dispersed, it survives well, and it infects diverse tissues. So you got a lot going, it's got a lot going for it. There's another common source of inoculum, Pachysandra, right? Isn't that, there was a report in 2011 that Pachysandra can be infected. So you got now that source of inoculum in the landscape. Cultural practices may have limited value. I mean, I'll modify this as the work with mulching goes forward. And fungicides, I think they're useful but not sustainable or desirable. But they're, I work with them, they're sometimes necessary. Another principle that I think a lot of us agree on in this field is that genetic engineering would probably preserve the qualities of the best varieties. You might arrive there through conventional breeding, and that's great. But if you have a particular variety that's got certain qualities, the way to retain that variety is probably to introduce the least amount of genetics possible. And that is a genetic engineering. Well, not necessarily. You could get there through backcrossing, but genetic engineering is more likely to get there. All right, the research proposal that's being discussed by my colleagues here from the USDA is gene silencing, and I was really excited to hear that. So Joanne, are you here? She's here somewhere. But yeah, there's, okay, hey Joanne. So they're talking about using a gene silencing approach. I won't explain the process, but I was like, right away I was like, yeah, that sounds like a really good idea. I think what they're doing is very likely to be worth doing. You can only know if you try. There's no guarantees in any research program, but it's a really good choice what they're talking about doing. We don't eat boxwood, right? Do we eat boxwood? I mean, okay, I really need you to shake your head, so thank you. I don't know, but I assume nobody eats boxwood. That's good, that's good. Then you don't have the food safety considerations, right? Transgene flow is something that, some sort of risk assessment about this. Remember, transgenes are genes that are from outside the breeding pool that have been introduced to the breeding pool by human intervention. Transgene flow, you would have to have a risk assessment as part of the approval process, but I sort of looked into it a little bit, and so as I think a lot of you already know, the common boxwood originates from Western Asia, Southern Europe, and Northern Africa. And so there is probably where I would go for the wild relatives. The only wild relative of Buxus that I could find was Buxus puertoricensis in Puerto Rico. And when I don't imagine boxwood would do at all well in Puerto Rico. So, you've got possibly a situation where risk to wild relatives is limited, which is another thing in favor of this as a possible pathway. In fact, I would, if the funds were available through USDA, I would do it, even if you were, just to have it, in the hopper. It's my opinion. But these are some of the bridges you'll have to cross. I do want to say the talk from Longwood Gardens, very good point. So we've got collections of plants that are non-GMO. So we would your risk assessment, would have to include how do we protect those plantations as well. But so it's not just about the wild relatives, but certainly wild relatives were on my radar before I came to this meeting. It is possible, if you were concerned about pollen flow, it is possible to engineer plants to not produce fertile pollen, as you see here. And I think there's technical ways to address challenges of all sorts. Going forward, here are a few bits of advice I'd say, and maybe a few points. One is, it's really important, if you go down this path, it's really important to engage the public early and often. And I think you want a process that is multi-stakeholder. You want people in the room that don't like genetic engineering. I think I would probably opt to include people that are willing to consider the possibility. maybe not a person who says, never, no way, over my dead body. That I don't know that degree of opposition is useful, but I would definitely want people that, from environmental organizations perhaps, that are presently opposed but willing to consider the possibility if it met certain conditions. But make it multi-stakeholder. Have them involved in the research all the way through deployment. They might give you good ideas, but you need them to be able to represent the program to the public if it comes to that, if you actually come to the point where you have a genetically engineered boxwood. And I think you have to build public trust. This engaging the public is one way, but also make sure you have boxwood sale systems down the road that are transparent. I mean, proudly label it as genetically engineered, let people know so they can make their own choice, and make, have traceability incorporated. The food industry is really moving fast towards traceability. So your industry will too, either way, but you would want that for genetic engineering. And I want to finish with a few pictures that I think are really important. This is a family suffering, whose cassava in Africa is suffering from this very virulent complex of viruses. There's a healthy, apparently healthy, but maybe infected, who knows, very sick. There are the cassava roots that have been dug up. Those that have been produced have these dark necrotic areas. It's a big deal. This is a big deal. And the only genetic solution to one aggressive component of this virus complex is genetic engineering at this point. And it's done by an African. And here are the traditional 2 varieties loaded with symptoms. Here are some of the engineered varieties. Not every engineering event, engineering insertion works. And here are some that did, and they look great. And that was done by Africans. I think they had Western collaborators, but I'm not going to tell them, those African scientists, they shouldn't do it. This is bacterial wilt of banana, so there, that pseudostem was cut open. Look at all the bacteria just oozing out of the xylem, the water conducting tissue. So it's going to lead to wilt. I mean, you're going to get wilt with that level of infection. And worse, the pathogen splashes to the fruit and rots the fruit. The only resistance I've seen in any scientific literature is GMO, genetically engineered with a single gene from pepper. And it involves African scientists. So it isn't all about Monsanto. This topic really gets conflated with Monsanto, and I'm not here to defend Monsanto. They've done some things I object to, but it's not all about them. It's really about a much wider set of possibilities. This is a completely African project. Oh, there might have been one Australian scientist. These plants were left, corn plants were left in the greenhouse for, I believe it was 3 weeks without water. 3 weeks. And the one on the left is engineered, the one on the right isn't. The corn from the engineered plant is on the right this time. And there it is. Hardly any, there's really no harvestable corn here on the left. So, and that was largely, again, an African project. So I think, last slide, this is it. Is that, it's just, to me, I think the most important thing is genetic engineering is a set of tools that can be useful. We have to truly and fairly evaluate the risks, but we really should do the evaluations in all cases on a case-by-case basis, not, for me, not across the board because it's genetic engineering.
So when you were talking about the genetically engineered plants, you didn't talk about if they were sterile or not as a result.
Right. Yeah, so is that your question? Yeah, sure. So there's a lot that has to get left out on the cutting room floor just for time's sake. There's a technology that I think you're referring to called the Terminator technology, and that was developed in actually by Monsanto. Oh man, I think maybe early 1990s. I can't remember exactly when. And what it was genetic technology modeled on natural phenomenon, by the way. So, but it was engineered by Monsanto. And what it does is if that terminator gene is in your seed, then it will flower and produce a crop of grain, but then those seeds from that grain, they'll look fine, but they won't germinate. And so it was called the terminator technology. That received such a global outcry of objection that it was never commercialized, never used. The reason they did it, I think there were a couple reasons why they did it. One is, it gives them control over the use, right, and patent protection. But the people worried about smallholders, right? And what about a smallholder who's saving seed and used to that, done it all their lives? The other actual advantage is it actually would prevent transgene flow, which I think was a potential advantage. But it was never commercialized. It still sort of reverberates through the media, but I've looked into it and I've seen only indications in credible sources of that it's never been commercialized. So, yeah.
So say I have an 18th century corn that I want to keep at 18th century.
Right.
And then Monticello is growing a genetically modified corn. And we have cross-pollination. Will I lose my 18th century straight species forever? Is that the big concern here?
Yes. Yeah, you're talking about transgene flow into your 18th century variety, right?
Right. I mean, isn't that the big concern, that the genetically modified is going to eventually pollination, cross-pollination, et cetera, could change those straight species that we want to maintain,
Right.
and you'll just lose those straight species and not be able to maintain it?
Yeah, sure. That's a good question. So when we, we're in the topic of transgene flow, I think if you want to pick a reason to object to genetic engineering, that qualifies. I think I'm always looking for ways to solve problems like that, but I think I legitimately can respect that somebody might have the position of, no, I don't want any transgene flow, because it introduces into the wild, because it introduces not risk but uncertainty. Okay, with respect to organic crops or heirloom crops, that would be that you're going to get some transgene flow if you have a very nearby genetically engineered crop. So I think the way that Europe actually does cultivate, there is some cultivation in Spain, I think Portugal, of genetically engineering, and they actually created a threshold of 0.9%. So 0.9% receiving if your crop had 0.9% or less of that trait, that genetic engineering trait, it would still be saleable. That may not be acceptable to you, so I think in that case, if I were you, I might want to work with my neighbor and to see if you could. Okay, maybe that's not going to work in this case, but I mean, some of the solutions that they studied in Europe include how far away do you have to be to get to a reduction below 0.9%, and it's not that far actually. It turns out. How much of a time separation in flowering between my variety and yours would reduce the risk? So there are ways to address the risk, but if your intention was zero, I would I don't think it's possible to get there in corn, because it is open pollinated.